Hey everybody, it's Mr. Fabeck here, and this is Fabiology, and we're going to talk about microscopes today. So, microscopes have changed over time. I love this comic. I think it's hilarious, right? It's a mammoth. Obviously, that microscope is doing its job, but very, very primitive. But now we have very, very complex ones. So, here's our topics for today. I can identify the parts of a microscope and explain their function. I can demonstrate the correct way to handle and use, and when we say use, we're talking about focusing, operating, maintaining safety, all of that, and also preparing a slide to view. Now, we're going to talk about the functions of how the slide will work and where it's going and how to kind of manipulate it. We'll talk about how to make a wet mount slide another day. I can calculate the total magnification on each objective lens. Now, it sounds difficult, but really, just some quick math, and we'll get right through it. So let's just get going. So we're looking at compound microscopes. Those are the modern ones. There's a variety of different ones out there. You can Google some images and see a whole diverse field. But they have changed from old, very old ones. If there's anybody you should remember about microscopes, it's Anton van Leeuwenhoek. Leeuwenhoek there in 1674 sort of had a monopoly on microscopes. That was it. It's very simple. You just had like basically a bead and a needle for the specimen, and he would hold it up to his eye and was able to see a pretty powerful image, which is very nice. He's kind of considered the father of microbiology at times because he was looking at pond water, bacteria, blood, all these really microscopic things that not a lot of people were able to do. And he kind of kept all those microscopes to himself, or at least the best ones, so that he could, make a little bit more money. The middle one there, that's our microscope that we're going to focus on. That's the modern day one that you see throughout high schools. And the other one is the scanning electron microscope. So it actually vibrates electrons, okay? It's actually going to vibrate those electrons, and it's going to produce these really amazing detailed images. So really check them out. Google image like parasites and bugs under electron microscopes. Oh, super cool stuff. So let's go with ours. So our microscope, especially a compound one, is generally just a very powerful magnifying glass. If you think about it, we're just using lenses to see things up close. So let's start with the top, the eyepiece. So we have the eyepiece here. That can actually rotate. Sometimes there's actually a needle inside of it. But the eyepiece is what you look through for seeing the specimen. You look down that body tube. And the eyepiece has a magnification of 10x, okay? So if we were looking just through the eyepiece, it's 10 times bigger than what it normally would be. The stage and the light, these can be your friends, okay? So some microscopes have a mirror that you have to manipulate the light to go through the aperture. Most of them just have a general switch. And the stage is just like you would go to the theater to watch something. The stage is what you want to look at. It's where you put your microscope slide and your specimen. Now, if you ever have trouble seeing your specimen through the microscope and through the eyepiece, sometimes adjusting the light source is what can really help get better, more detailed images. And how do we do that? Well, we use our diaphragm. The diaphragm, there's two different styles here. Some have a rotating one, some have just this slide. But ultimately, as you can see, there's those holes. It's changing the aperture, just like a camera, and we're changing the amount of light that comes through. And so by reducing the amount of light, sometimes we get a more detailed image. So this is your friend. If at any point you're having trouble focusing, try adjusting the light and see if that helps. Stage clips. Okay, usually there's two of them. They're metal. They hold things in place. Some stage clips are actually really nice where you can lock your slide into place, and then there's a little twist bar so you can actually move it back and forth to see the whole field, okay? See what you're looking at. Objective lenses, there's 4 total. Now that's including the eyepiece, okay? The first one is the smallest one. That's usually denoted in a red color. We call that the scanning one because that's what you can scan your whole field on. It's the one that sees the specimen the furthest away, and that's 4 times what it would be. So quick math, if we want to find the total magnification, it's 10 times 4. Now where did I get 10? That's the eyepiece. The eyepiece is 10x, the scanning is 4x, 10 times 4, 40 times as big as what it normally would be. Then you can rotate your to a higher objective. We have the low power or yellow. That's 10 times bigger, so 10 times the eyepiece times 10, right? 10 times 10 is 100. The total magnification on yellow is 100x. And high power, this is blue. This is 40x, so 40 times 10 is 400. There are some microscopes that do oil immersion that would be 100x, and they can even do even bigger. So those ones are very, very cool. But again, we're just going to focus on the general ones that you see. So total magnification, eyepiece times the objective. Very simple. So the coarse adjustments and fine adjustment knobs can be found on the side. The coarse adjustment knob moves the entire stage. Now this is important. When you're using the coarse adjustment knob, you're only using it on the red. Do not use it when you're looking at anything through the yellow objective and the high-powered blue objective. Because it moves the stage, it can crack the lens and it can crack your slide and damage the microscope, and everything in your experiment could be lost. Okay, so do not use the coarse adjustment one. What you should use after you focused in on red on the scanning is the fine adjustment. Once you switch to yellow and blue, just use the fine adjustment knob. It actually will make it much, much easier to focus and a lot more clear. The nose piece, that's where all of these objectives are. They just click into place. You can rotate it around. If you can't view through the nose piece, then you have, oftentimes you're not clicked all the way. You should hear a clicking sound and the image should be clear. If it comes out black, your objectives aren't in place. Here's how you carry it, right? We want to carry it by the arm and that base. We're all about the arm and the base when we're talking about microscopes. This way you will always ensure that you're carrying it so that the objectives don't get damaged and that you don't trip and lose control and all that stuff. It's just a very, very easy thing to get in our brain and how we transport our microscopes. So that's it. That's the basic parts of a microscope. Here's a diagram. You can see other diagrams online, so I encourage you to do that, to check out various amounts. There's one thing that I would do personally on this. We can see we have our nose piece at number 2, right? And we have 3 different sizes. Okay, I would add a line here. I don't know why I didn't do that, but there should be another line in here to label what is that middle level objective, right? And also put the total magnification, even including the eyepiece on there, and that will help you practice and master these skills so that you can go into any lab and view any specimen and see all that sweet stuff that's in pond water and all that gross things that's crawling all over this world. So thank you, and we'll see you in class. If you have any questions, don't be afraid to ask. Bye, guys.